Good morning. Today I will start to talk about one of the accessory organs that found in the digestive system, which is the liver. Our objective for this lecture to identify the liver in the glass slide or regular slide, to understand the major liver functions, and also to follow the flow of the blood and bile through the liver. When we consider the function of the liver, the main function, we have one of these functions, which is a gluconeogenesis, which convert amino acids and lipids into glucose. The other function, which is detoxification, and this is very important for the removal of drug and toxin from the blood, and usually they're released through the bile. The third function, which is the storage of the glycogen and triglyceride. The other function, which is production of the plasma protein, which are the fibrinogen, which is very important in the blood clotting, prothrombin, and albumin. Finally, we have the production of the bile, which is the exocrine function of the liver. Liver is the largest gland in the mammal. One of the largest glands found in all mammals is the liver. Usually they are uniform histologically across the species. By this I mean the liver of the dog is similar to the sheep, goat, cow, horse, even human. The liver is multitask. They have secretory, protective, and synthetic function. And very important, it's capable of considerable regeneration. If the liver we lost all that area of the liver have considerable capability of regeneration. It could regenerate the whole structure of the liver, and this is the only organ in our bodies that can able and have ability to regenerate. The liver, in a gross histological slide, is covered from outside by simple squamous epithelium, which is the mesothelium. Beneath this epithelium, we have thin layer of a connective tissue, which is the Glisson's capsules. Usually, these capsules send trabeculae inside the liver, and these trabeculae help in the dividing the liver into classical liver lobules. The cells of the liver is called hepatocyte. Are each one of these is the hepatocyte, and you can see the general arrangement of these hepatocytes are plate of cells. See here, this is the plate of cells. There's anastomosing between these plate of cells, and between these spaces we find sinusoids, which are large blood capillaries. Okay, so from outside we have the simple squamous epithelium, the mesothelium. Beneath it we have the connective tissue, and as I said, they send trabeculae, divide the liver into lobules. Each one of these cells is the hepatocyte, which is the main parenchymal cells of the liver. These cells is form plates of hepatocytes. Between them there is anastomosing. The space between the hepatocyte, which is the sinusoid, and usually this sinusoid is filled with blood. Hepatocyte, as I said, they have several functions, so they have well-developed rough endoplasmic reticulum for the process of the protein synthesis. We have small endoplasmic reticulum and lysosome and proximers. All of these very important for the process of the detoxification and for the lipid metabolism. Inside the liver, as I said, there's a storage of the glycogen and lipid droplets. See here, this is the simple squamous from outside. This is the connective tissue. This is the plate. This is the plate of the hepatocyte. And the space between the hepatocyte, as I said, this is the area of the sinusoid. So these spaces between the hepatocyte, this is the area of the sinusoid. And usually these spaces between the plate of the hepatocyte. So we can see here this hepatocyte. There is anastomosing with adjacent hepatocyte. The space between them is the area of the sinusoid. As I said, this area will be large blood capillaries with large fenestration and discontinuous of basal lamina. As I said, from outside we have the mesothelium and we have the capsule, and these capsules send the trabeculae, and this trabeculae, as we can see here, goes interior and divide the liver into classical liver lobules. Each one of these is called the classical liver lobules. In the central of these lobules, we have the central vein, and we can see here this is the area of the portal area where the blood vessels, hepatic artery, and the portal vein send the branches to this area here. Each one of these is called classical liver lobule, and the general arrangement is the hexagonal arrangement. As I said before, the hepatocyte is the principal cells of the liver, so that's why we call it parenchyma, whereas the connective tissue is called the stroma. As I said, these cells have well-developed smooth endoplasmic reticulum, rough endoplasmic reticulum for synthesis and detoxification function. As I said, also it's the site for the carbohydrate storage, also for the lipid storage. We can see here, this is the hepatocyte with the Golgi apparatus, lipid droplet, well-developed rough endoplasmic reticulum. And we can see here, between the hepatocyte, there is the sinusoid, which is the area where we have maximum contact between the hepatocyte and the blood. For the classical liver lobules, as I said, we have plate of cells. We call it the hepatocyte, which is the parenchymal cells. Usually these cells can be binucleated. By this I mean instead of having one nucleus, they have two nucleoli, and the amount of DNA will be more than the normal, which is 2x of the normal diploid DNA. So sometimes we call it polyploid, which is the several times the normal DNA amount. Now we'll talk about the blood supply for the liver. There's two main arteries, blood vessels that enter the liver. We have the hepatic artery, which is a branch from the abdominal aorta, and we have also the portal vein, which return the bloods from the intestine. These two bloods enter the liver and leave through one vein, which is the hepatic vein. And usually this hepatic vein joins the caudal vena cava, and that goes directly to the heart. So you can see here, this is the portal vein that's coming from the intestine, and this goes to the liver and then branches and will be collected through the hepatic vein. The importance of the hepatic artery, which is the main blood supply for the liver, provide the nutrition and the oxygen for the heart. The liver as an organ, like other organs in the body, need oxygen and nutrient, and the hepatic artery usually provide this supply. Usually the blood that enters the liver, 80% is coming from the portal vein, whereas from the hepatic artery, only 20%. As I said, the drainage will be through the hepatic vein, which will go through the caudal vena cava to reach the heart. As I said, we have the classical liver lobules. As I said, we have capsule from outside, send it trabeculae, and this trabeculae surround the plates of the hepatocyte, and this will result in the formation of the classical liver lobules, and usually it's hexagonal arrangement. It's with six. See these here? We call it the hexagonal arrangement. As we can see here, we have a plate of the hepatocyte. Between the hepatocyte, we have the sinusoid. They collect at the center through the central vein. Around the periphery of each classical liver lobules, at 2 or 3 angles only, we have this structure here. This structure is called the portal area. Sometimes it's called portal triad or portal area. So we have 3 names for the same area, which is the portal canal, which is called also portal triad or portal area. At this area here, we have branches from the portal vein, which is coming from the intestine. Also, we have branch from the hepatic artery. Sometimes we can see lymphatic vessels, but very important, we see branch from the bile duct. So that's why they call it triad, because we have hepatic branch from the hepatic artery, we have a branch from the portal vein, and we have a branch from the bile duct. If we increase the magnification for this area here, we can see here, this is the vein. See here, this is the wall, the tunica intima with the tunica adventitia. Here we have the artery. We have the tunica intima, media, adventitia from outside, and we can see here, this is the lymphatic vessels. But look here, you can see the epithelium is simple cuboidal epithelium. As we know that the vessels usually they have simple squamous epithelium, whereas this one here we have the cuboidal epithelium. So this one here we have the portal vein, this is the hepatic artery, and this is the bile duct, and we differentiate between them depend on the lining epithelium. If it's cuboidal, then it's bile duct. If it's squamous epithelium, depend if we have tunica media with the smooth muscle, then it's the hepatic artery. If all you can see the simple squamous epithelium or tunica adventitia with large vessels with a lot of red blood cells inside it, then it will be the portal vein. We can see here this tube with simple squamous with a tunica adventitia, but we don't see any red blood cells. This is the lymphatic vessels. So again, this is the classical liver lobules, classical hexagonal arrangement. At 2 or 3 angles, we have the area of the portal area. At the center here, we have the central vein, but we could not see like through vein. We could not see the tunica intima or media or adventitia. It's just a space where the blood will be collected. So at this area here, we have the classical liver lobule. It's a plate. You can see this is a plate of the hepatocyte. Between these hepatocyte and this area here, we have the area of the sinusoids where the blood will be collected, and there will be a maximum contact between the blood cells and the hepatocyte. These sinusoids usually they have this continuous endothelial cells and a lot of spaces between these cells. As you can see here, this is a plate. Sometimes we have area of this continuous, so we can see that there is some junction between the different sinusoids. It's not through one space. There's area of disruption of these hepatocytes, and this is allows maximum and mixing of the blood. As I said, the liver, they have endothelial cells as the lining with a large fenestration. we have additional cells that found in the sinusoid. These cells are called the Kupffer cells, which are phagocytic cells, and usually these cells are monocyte. In the blood, we call them monocyte. Once they travel and migrate toward the liver, their name will be changed. Usually we call them as the Kupffer cells. These cells, we could find them in these sinusoid or inside the sinusoid, between the endothelial cells or inside these sinusoid. So this is a little bit, the image is magnified. This is the area in the center where we have the central vein. We have the plate of the hepatocyte. Between these hepatocyte, we have the sinusoid. All of this area, see this area here where we have the disruption of the plate. So there's mixing between the blood that found at the sinusoid at this area here and the sinusoid at this area here. For the blood flow, usually we have branches of the hepatic artery, the portal vein, and usually their blood will go directly in the sinusoid from this direction, from the periphery toward the central vein. And this is similar also for the hepatic artery. Sometimes there's area of disruption in this area, so there will be a mixing between the blood from the venous and the arterial blood supply. See this area here? This is the area of disruption I was talking about. So this is the area of the portal area where we have the portal vein, hepatic artery, and the bile duct. And we can see here they send their branches, and these branches reach to the area of the sinusoids. See here, this is the central vein. We can see the lining epithelium, which is the simple squamous epithelium. And we can see here we have the area of the sinusoids. And as I said, we have this cell here that's found between the endothelial cells or inside the sinusoid, which is the Kupffer cells, which is phagocytic monocyte lineage cells. If we use a special stain, we can stain these cells with the carmine red. This is a special stain, and you can see all of these cells here. These cells are the Kupffer cells. As I said, they're found between the endothelials or sometimes inside the sinusoid. As I said, between the hepatocyte, this is the hepatocyte, between the hepatocyte plates, we have the sinusoid. As we can see here, this is fenestrated. This is a large space, and we can see here the microvilli of the hepatocyte. So this will allow a maximum contact between the blood and the hepatocyte. As I said, the hepatocyte is going to modify the component of the blood. So we need a maximum contact between the blood and the hepatocyte. See here, all of these area, this is the area of the sinusoids. And inside it, we can see the microvilli of the hepatocyte. For the bile flow, it's opposite of the direction of the blood. As I said, the blood will flow from the periphery of the classical liver lobules from the portal area or the triad toward the central vein. Here will be the opposite, from the central vein toward the portal area. The bile, which is the exocrine secretion of the hepatocyte, usually secreted into bile caniculi, which is area of the intercellular space between the hepatocyte. Then this bile is going to flow from these canals, the canals of Herring, small bile duct, toward the edge of the liver lobules, and then into the bile duct which is located in the portal area. So at the beginning here, this is the area of the sinusoid, and you can see this is the area of the fenestration. As you can see here, the area of the microvilli. The blood cells will be found at this area here for the maximum contact. But here, at this area here, we'll have two adjacent hepatocytes. Between them, there is a junction. But we can see at this area here, we have this area of the separation in the plasma membrane. This space here between the hepatocyte, which is the bile caniculi. And usually these hepatocytes send and excrete their secretion at this space here, which we call it the bile caniculi. And these bile caniculi, as I said, will be collected and reach to the level of the bile duct that found in the portal area. So the direction, as I said, for the blood will be from the periphery toward the central vein, whereas for the bile will be from the area of the central vein toward the portal area. So this here, this is the flow of the blood. Whereas the bile from this area here. So the bile is going from this area here toward this canal here, which is the bile duct that are found in the portal area, whereas for the blood is going from the portal area here toward the central vein and will be collected in the central vein. For the bile function, the main function is the excretion of the cholesterol, the phospholipids, the bile salt, and conjugate bilirubin and electrolyte, and also play a role in the fat absorption, and very important in the excretion of drugs and heavy metals. Our body gets rid of these drugs and heavy metals through the bile secretion, which goes from the liver through the bile duct to reach the small intestine. One condition that can affect the heart called the cirrhosis. Normally, as I showed you before, that the amount of connective tissue is very minimal, and the only classical liver lobules that I showed you before, we can see it only in pigs. They have very good amount of connective tissue. However, in other animal species, there is a limited number or amount of the connective tissue. Sometimes the condition which is called cirrhosis, there is increase in the amount of the connective tissue. This is special stain for the connective tissue, and we can see here, this is abnormal. This condition here is called cirrhosis. Sometimes it happens with people with heavy drinking of the alcohol. Other important structure we have, which is the gallbladder. This is very important because it stores and releases the bile. The bile is stored and released into the duodenum through the gallbladder. Usually the gallbladder consists of mucosa and submucosa, and we have smooth muscles, and we have the area of serosa. Usually the mucosa, which is lined by simple columnar epithelium, the layer just below it, which is the lamina propria, usually will be fused, and we could not separate it from the underlying layer of the submucosa, which is the dense, irregular connective tissue. Beneath these two layers, we have an area of the smooth muscles, and as I said, finally we have the area of the serosa. We can see here, this is the area of the epithelium, and this epithelium here, which is simple columnar epithelium, this is the area of the lamina propria, and we can see here just beneath it, we have the area of the submucosa. As we can see, there's no true separation between the lamina propria and the submucosa. Down here, we can see the area of the smooth muscles. So again, this is the epithelium, which is the simple columnar epithelium, and just beneath it, we have the area of the lamina propria, which is loose connective tissue. For the diseases that happen in the gallbladder, the most disease happen, which is the gallstone. So this is like gallstone concretion, and usually these gallstone concretion usually they are cholesterol crystals. Sometimes they use the ultrasound waves to destroy these stones, and sometimes they open the abdomen and remove the whole structure, which is the gallbladder. As I said in the beginning, we have the classical liver lobules, which is the hexagonal arrangement. As I said, in 2 or 3 angles, we have the portal area. We have some alternative organization or model of the liver organization. We have something we call it the portal lobules, and this consists of the centered and portal area and is defined by 3 adjacent central veins. So this is here in the central, we have the portal area, and we have at the 3 angles, we have the central veins. And usually this one here describes the flow of the bile toward the portal area. This one here, we call it the portal lobules. As I said, it defines the flow of the bile into the bile duct. Usually it's triangular in shape. At the 3 angles, we have the central vein, and at the middle here, area here, we have the area of the portal area. So again, this is the hexagonal arrangement, which is the classical liver lobules. This is the portal lobules, which is triangle in shape. At each angle, we have the central vein, and at the center here, we have the portal area. As I said, it describes the flow of the bile toward the bile duct. The other one, which is the hepatic acids, usually define the blood flow. Usually define how the blood is flow in the liver, and usually it's composed of the diamond shape. This one here, this is diamond shape, and we can see at 2 angles we have 2 central veins and 2 or the other 2 angles we have the portal area, and from this area here toward this area here describe the blood flow. This is very important in case of the toxicity. For example, if animal or human ate some toxic materials, this area here, which is around the portal area, we have the portal vein that's coming from the small intestine. So this area here will be the first area to expose to toxin. So these maybe have the most damage due to the toxin. As we go far toward the central vein, the toxin effect will be reduced. Okay, so this one here describes the blood flow and which area will be more affected by the toxin, and in the other hand, which area they have the richest metabolic and nutrient and oxygen, because as I said here, we have the portal vein which return blood from the intestine, which is carry the nutrient, whereas the hepatic artery, a branch from the hepatic artery, will carry the oxygen and nutrient to the liver structure. So this here again, this is the diamond shape. At 2 angles we have the central vein, at the 2 periphery we have the portal sinus. As I said, this describes the blood flow, and there's gradient. So at the beginning here, this area here, we have the richest blood supply. As we go down, There's reduction in the amount of the oxygen and nutrients. However, this area here will expose more to toxin compared with to the area around the central vein. So this is the lecture about